Hello. Today I'm going to be talking about homeostasis and protein mediated transport. Let's start by defining homeostasis. Homeostasis is the body's ability to maintain a stable environment despite external changes. We can compare this to a thermostat in a house, ensuring that the temperature stays constant regardless of the weather outside. Now, let's see how homeostasis works in the body. A classic example is blood sugar regulation. When you eat, your blood sugar rises. In response, the pancreas releases insulin, which prompts cells to take in glucose, lowering the blood sugar back to normal. To understand this process better, let's explore the components of the reflex control pathway. It starts with a stimulus, like the high blood sugar, this triggers receptors, such as those in the pancreas, to send signals via sensory neurons that make up the afferent pathway to the control center, often the brain or the spinal cord. The control center then signals through neurons that make up the efferent pathway to the effectors, which have the means to correct the change from the stimuli. Within the control center, two feedback loops come into play: negative and positive feedback. Negative feedback loops work to reverse the initial change, such as insulin lowering blood sugar. Positive feedback amplifies the change. An example of this is during childbirth, when contractions begin to intensify until delivery. There can be competition between feedback loops, which results in a hierarchy between them, with the balance in the brain being the highest priority. Following that example, we can talk about the example of negative feedback with body temperature regulation. When it's hot, sweat is used to lower the body temperature, and when it's cold, the blood flow in the upper and lower extremities of the body will decrease, allowing for more of the blood flow to be inside the body to keep the temperature of the vital organs at a good temperature. Another example is loss of blood. If you lose blood, the blood pressure in your body will decrease, which will result in vasoconstriction of your vessels, and your heart rate will begin to run higher. Another example of positive feedback is seen with blood clotting. When cut, the body will send platelets, which will signal for more platelets as they arrive. Now let's shift gears to protein-mediated transport, which are crucial for moving substances across cell membranes. First, we have diffusion through channels, where specific ions are able to pass through protein channels from areas of high concentration to low concentration. Voltage-gated channels will open when the voltage of the membrane changes. The concentration of ions in the intercellular fluid create the voltage. They are sensitive to charge and cause the channel to open for a specific ion. The ligand-gated channel open when the ligand binds to a specific location, allowing for ions to pass through, once again following the concentration gradient. Mechanical gated channels open when changes such as pressure, touch, or temperature occur. Leak channels open and close normally depending on the concentration gradient, and which contributes to the cell's electrical potential. Next, we have carrier-mediated diffusion, which involves proteins that bind to specific molecules, carrying them across the membrane, once again following their concentration gradient. Finally, active transport uses energy, often ATP, to move molecules against their concentration gradient, ensuring substances like nutrients are absorbed even when their concentration outside the cell is low. In primary active transport, energy is directly expended, usually in the form of ATP, to pump molecules across the cell membrane. They are specific like the channels in the carrier protein. One of the most common examples is the sodium-potassium pump. This pump actively transports sodium ions out of the cell and potassium into the cell against their respective gradients, using ATP as an energy source. You can see here that it takes 3 sodiums out and 2 potassiums into the cell. Secondary active transport utilizes the energy stored in an electrochemical gradient created by the primary active transport to move molecules against their concentration gradient. This process involves the co-transport of 2 or more molecules or ions, with one moving down its concentration gradient while the other moves against its concentration gradient. For instance, the sodium-glucose co-transporter and the intestinal cells use the sodium gradient created by the sodium-potassium pump to transport glucose against its concentration gradient into the cell. When both molecules go in together, it's called symport, and when 2 molecules go in and out opposite of each other, it is called antiport. With all these different types of protein-mediated transport, there is a max rate that they can allow ions and molecules to pass through. Even if you increase the amount of solutes, it will only diffuse them at its max rate. Increasing the solute would essentially leave them saturated. In conclusion, homeostasis maintains stability in the body through feedback loops, while protein-mediated transport ensures vital substances move in and out of the cells efficiently. Thank you for joining me on this journey through homeostasis and cell transport. Hello and welcome. Today we'll be going over the topic of osmosis and resting membrane potential. Starting with osmosis, osmosis is the process by which solvent molecules, usually water, move across a semi-permeable membrane from an area of lower solute concentration to an area of higher solute concentration. In order to equalize the concentration of solute on both sides of the membrane, this movement of solute continues until equilibrium is reached, where the concentrations of solutes are equal on both sides, or until the osmotic pressure difference prevents further net movement. Osmolarity is a measure of the concentration of solute particles per unit of volume, typically expressed as osmoles per liter. It takes into account all solute particles present in a solution, regardless of their chemical nature. Molarity, on the other hand, is a measure of the concentration of individual molecules in a solution, expressed as the number of moles of solute per liter of solution. So the molarity focuses on the concentration of a particular solute. Osmolarity considers the concentration of all solute particles in a solution. Tonicity, however, refers to the effect a solution has on cell volume. It is determined by the concentration of solute particles inside versus outside the cell. A solution can be isomotic, meaning it has the same osmolarity as the cytoplasm, but hypotonic to the cell if it has a lower concentration of impermeable solutes, such as sodium, compared to the cell's cytoplasm. Impermeable solutes are solutes that are not able to pass through the cell wall, where permeable solutes can. In this case, water will move into the cell via osmosis, causing it to swell and possibly burst. Conversely, a solution can be isomotic and hypertonic to the cell if it contains a higher concentration of impermeable solutes compared to the cell's cytoplasm, leading to water moving out of the cell and causing it to shrink. Another example would be a solution that is hyperosmotic and isotonic. Since there are equal amounts of non-permeable solutes, it is isotonic, but since there is dextrose, the solution outside the cell is more concentrated, making it hyperosmotic. Since dextrose is permeable, it doesn't affect the tonicity of the cell, and it can balance itself out without needing to move water. The resting membrane potential is primarily established and maintained by selective permeability of the cell membrane to ions, especially potassium and sodium, and the activity of ion channels and pumps. One of the pumps that is commonly talked about is the sodium-potassium ATPase pump. This pump actively transports 3 sodium ions out of the cell and 2 potassium ions into the cell against their respective concentration gradients. Using ATP, this pump creates a higher concentration of potassium ions inside the cell and a higher concentration of sodium ions outside the cell, establishing a concentration gradient for both ions. A big part of ion movement is through the use of protein channels that we discussed in the last video. As a reminder, we have our gated channels, which are closed until they are activated, and our leak channels that are just open most of the time. The one I want to focus on here is the voltage gated channels. As you can see in this diagram, as the ions pass through the membrane, the charges change. If the positive charges decrease on the outside, the spring-like helix will move up, allowing the activation gate or the lever to open. As the ions move out of it, the ball-like inactivation gate will come up and close it off quickly. Another factor that changes the resting membrane potential is permeability of the membrane to ions. For example, if we were to open more potassium channels, it would increase the potassium movement, making the cell more negative. Conversely, changes in sodium permeability can also influence the resting membrane potential. The Nernst equation is a mathematical equation that gives us an idea of the equilibrium potential of an ion to cross a membrane when the same type of ion has crossed it, driven by the concentration gradient. It's expressed as E equals 61.5 millivolts over Z times log ions inside over ions outside. E is the equilibrium potential of ions. Z is the charge of the ion, and the ions inside over ions outside is the concentration of the ions outside and or inside and outside the cell, respectively. The driving force for ion movement across the membrane is the difference between actual membrane potential and the equilibrium potential for that ion. If the membrane potential is more positive than the equilibrium potential for a positively charged ion like sodium, the driving force will be inward. Conversely, if the membrane potential is more negative than the equilibrium potential for that ion, the driving force will be outward. Hello and welcome. For this video, we're going to be talking about action potential. And so action potential is a fundamental process and is crucial for the transmission of signals within and between neurons. Starting with the characteristics of action potential. Action potential, once initiated, an action potential will spread along the neuron's axon without decreasing in size, or it won't occur at all if the threshold is not met. Depolarization, the membrane potential rapidly becomes less negative and more positive, and this is during a rising phase of action potential. Repolarization, the membrane potential returns to its resting state after it depolarized. And then after it repolarizes, it actually hyperpolarizes. The membrane potential briefly becomes more negative than it was originally. The refractory periods are periods during and after the action potential where the neuron is less responsive to another stimulus. So, going a little more into detail, the rising phase of depolarization, a voltage-gated sodium channel or channels open in response to a stimulus, allowing a rapid influx of sodium ions into the cell. This rapid influx causes the membrane potential to become more positive and pass the threshold to initiate the action potential. The following phase, or repolarization, the voltage-gated sodium channels close and the voltage-gated potassium channels open. The potassium ions then flow out of the cell, causing the membrane potential to become more negative again, returning towards the resting potential. After that, during that, the potassium channels remain open for a brief period, causing an excessive outflow of potassium ions, which leads to this hyperpolarization of the membrane potential before it returns to normal. The absolute refractory period, going into a little more detail with that, during this period, sodium channels are inactivated and incapable of opening, which prevents the initiation of another action potential. And this is seen, this is the period as it's rising and as it's above the threshold, essentially. Once it drops down below and hyperpolarizes, this divot here, this is the relative refractory period, which follows the absolute refractory period, and potassium channels are still open and the membrane potential is hyperpolarized. And it's possible to initiate another action potential, but it requires a stronger stimulus since it's dropped below the original, where it was originally at. So kind of coming in, comparing this with graded potentials. Graded potentials are small changes in membrane potential that occur in response to a stimulus. They can be depolarizing, making it more likely to fire an action potential, or it could hyperpolarize, making the neuron less likely to fire an action potential. Where action potentials are rapid and large changes in membrane potential that occur when a neuron is sufficiently stimulated. The action potentials are the means by which neurons communicate and transmit signals over long distances. Graded potentials have their magnitudes vary depending on the strength of the stimulus, and the stronger the stimulus produced, the larger the graded potential will be. And action potentials, their magnitude are fixed. These don't change. Once it hits the threshold, it will always reach the same magnitude when the action potential is triggered. It doesn't change. The graded potentials are local events that occur in the dendrite and the cell body of a neuron. They don't send signal over long distances, and they lose their strength as they travel along the neuron. And whereas action potentials, they send a signal along the entire neuron, like we talked about before. Graded potentials can be initiated by various stimuli, including neurotransmitters, sensory stimulus, or spontaneous changes in the membrane potential. Action potentials are initiated when the membrane potential reaches the threshold level. Moving on to terms, we have capacitance, which refers to the ability of the neuron's membrane to store charge. It's influenced by the surface area and the thickness of the membrane. For example, we can see here as we go down the axon, the capacitance is higher where there is no myelin, but when you have these myelin sheaths, the capacitance goes down. The time constant represents the time it takes for the membrane potential to reach approximately 63% of its final value during charging or discharging. It's determined by the product of the membrane capacitance and the membrane resistance. The length constant defines the distance over which a change in membrane potential can send a signal with minimal decrease in power. And it's influenced by the membrane resistance and intra and extracellular resistances. At a chemical synapse, the presynaptic terminal releases neurotransmitters into the synaptic cleft, where they bind to receptors on the postsynaptic membrane, leading to a change in postsynaptic potential. So when neurotransmitters are released, action potential arrives at the presynaptic terminal, depolarizing it. This depolarization triggers the opening of a voltage-gated calcium channel, allowing calcium ions to enter the presynaptic terminal. Calcium influx leads to fusion of synaptic vesicles with the presynaptic membrane and release of neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane, causing ion channels to open or close. And depending on the type of neurotransmitter and receptors involved, the postsynaptic membrane may depolarize, changing the excitatory postsynaptic potential, or hyperpolarizing, which is the inhibitory postsynaptic potential. Depending on which one of these two potentials occur, it will, if the summed potential reaches the threshold, an action potential is initiated and then propagated along the axon. Hello and welcome back. For this last video, we're going to be talking about the sympathetic division of the autonomic nervous system, the parasympathetic division, and the somatic nervous system. We'll be talking about different things like where they originate, the number of neurons, the location of their ganglia, as well as neurotransmitters and types of receptors they have. So starting with the sympathetic division, the sympathetic division originates off of preganglionic neurons that come off of the lateral horns of the thoracic and lumbar segments of the spinal cord, starting from T1 to L2. The number of neurons is 2 in this pathway. We have preganglionic and then postganglionic. The sympathetic ganglia are close to the spinal cord and form chains along both sides of it. There are also prevertebral ganglia located anterior to the spinal column. Neurotransmitters used here, they use acetylcholine while in the preganglionic fibers, and while in the postganglionic fibers, they release norepinephrine. The type of receptors on these pathways include adrenergic receptors, which are alpha and beta subtypes. The sympathetic division is known for the fight or flight system of the body, which increases heart rate, dilates bronchioles, constricts blood vessels in non-essential organs, and releases glucose from the liver. The parasympathetic division originates in the brainstem and the sacral spinal cord. Similar to the sympathetic division, there are 2 neurons. We have preganglionic and postganglionic. The location of the ganglia for the parasympathetic are located near or with the target organs. The neurotransmitters used again is the acetylcholine, which is released from the preganglionic fibers, but different from the sympathetic nervous system. The postganglionic fibers also release acetylcholine. For receptors, the parasympathetic system has cholinergic receptors, which include nicotinic and muscarinic subtypes. The parasympathetic system is known as the rest and digest system, causing slowing of the heart rate, constriction of the bronchioles, and promoting digestion and conserving energy in the body. The somatic nervous system originates in the central nervous system in the motor cortex of the brain. There is one neuron in the pathway for the somatic nervous system, and it comes from the central nervous system to the skeletal muscles. There are no ganglia in the somatic nervous system like there are in the para and the sympathetic nervous system. The somatic motor neurons also use acetylcholine at their neuromuscular junctions. The somatic motor neurons for receptors interact with nicotinic cholinergic receptors on the skeletal muscles. The somatic nervous system controls voluntary movement of skeletal muscles, which is different from the other two, which are involuntary. The cholinergic receptors respond to the neurotransmitter acetylcholine. The subtypes are the nicotinic receptors found in the sympathetic and the parasympathetic ganglia, as well as on the skeletal muscles. The activation of these leads to depolarization for action potential. The muscarinic receptors found in target organs of the parasympathetic nervous system lead to various responses depending on the organ. So for the heart, for example, slowing the heart rate or increasing granular secretions. The adrenergic receptors respond to the neurotransmitter norepinephrine that are released by the sympathetic postganglion fibers. The subtypes here are alpha receptors, so you have an alpha 1 and an alpha 2 receptor. Alpha 1 receptors typically cause smooth muscle contractions. For example, vasoconstriction in the blood veins. And the alpha 2 receptors can inhibit neurotransmitter release. So you'll notice a kind of theme here. Odds excite, and then evens inhibit. The next subtype are beta receptors. So beta receptors, we have beta 1, beta 2, and beta 3. Beta 1 receptors are primarily located in the heart and increase the heart rate and its contraction strength. Beta 2 receptors are found in the bronchioles and blood vessels, leading to bronchodilation, allowing for more oxygen to be pulled into the lungs, and vasodilation. Beta 3 receptors are involved in fat breakdown. Moving on to mechanisms of action for these receptors. So coming back to the cholinergic receptors, when the acetylcholine binds to the nicotinic receptors, ion channels open, allowing for influx of sodium and depolarization of the postsynaptic membrane. muscarinic receptors activate G proteins, leading to opening or closing of ion channels, or the activation of intracellular signal pathways. The adrenergic receptors are activated by norepinephrine and lead to the activation of intracellular signal pathways through G proteins, resulting in physiological responses specific to the receptor type. So once again, there was vasoconstriction, bronchodilation, and then increased heart rate. Thank you for watching these videos, and we'll see you next time.